So thank you for coming. We're going to be discussing today ultra-low variant detection using the de novo sequencing method. And Nick, can you go to the next slide for me really quick? So all of the methods and tools that our speakers are going to discuss today are available on both Galaxy and Galaxy Pro. So Galaxy is a bioinformatics software platform that incorporates thousands of bioinformatics tools in an easy to use interface. And you can run those tools on public servers, which are accessible to anybody who has an account. And there you can access community help and interact with the very large and active Galaxy community. Galaxy Pro is a subscription-based service which manages Galaxy and provides this provides the Galaxy platform with validated tool sets and workflows, as well as increased privacy and security options, and has dedicated support. So I've provided the links where you can get more information on both of those platforms. Next slide. So today we are joined by two speakers who are going to talk to us about the method and how to do this in Galaxy. So Dr. Barbara Arbeithuber is a research group leader at Johannes Kepler University in Linz, and we'll be introducing the method and talking about how she has used this in her research. And Dr. Nick Stoller from Penn State is going to be discussing how do you access and use these tools inside of the Galaxy platform. So with that, I'm going to hand it off to Barbara to introduce the method.
Okay, so hello everyone and welcome to today's webinar, also from my side. First, as mentioned, I just want to quickly give an overview on the duplex sequencing method helpful in detection of rare variants. And if you go to the next slide, Nick, for me, please. The method was already published in 2012 by the group of Lawrence Loeb. But still, until now, it is one of only very few methods that is capable of measuring really low mutation frequencies. And the reason for this is that with duplex sequencing, you use double-stranded adapters that have a double-stranded random sequence, as shown here with this ends. And usually it has a size of 12 nucleotides. And next slide. So in duplex sequencing, you ligate these adapters with the random sequence to your fragmented double-stranded DNA of interest, and so you end up with a different random sequence on both ends of your double-stranded DNA, and each different DNA fragment has a different combination of such a random sequence. And only after the ligation of this double-stranded duplex adapters to your DNA, PCR amplification is formed. And because of the asymmetric nature of the adapters, when then looking at the sequence in 5', 3' direction, you end up with a different combination of this random sequence for the two initial DNA strands of your double-stranded molecule. So as shown here, with alpha and beta as examples for the random sequence, one strand has alpha beta combination, while the second strand has beta alpha. And then after those libraries are prepared, the end sequencing is performed. And next slide. when then analyzing the sequencing result, you first form consensus sequences. And this formation of consensus sequences is done independently for the two initial strands of your double-stranded molecule. So again, here the alpha beta family, and so you end up with two single-stranded consensus sequences representing the two initial DNA strands of your molecule. By doing this, you can already get rid of a lot of bias coming from late PCR errors or from sequencing errors. But still, if, for example, an error happened very early during PCR, or if you have, for example, a DNA lesion such as 8-oxoguanine, which often leads to G2T mutations, it would be present as a mutation in your single strand consensus sequence, because then it would be present in either the majority or all of the reads coming from the PCR product sequence for this strand. And usually a cutoff of around 70% is used for calling a variant. So only if a variant is present in at least 70% of the sequenced reads, then it is called as a variant. And so how can you then get rid of such false positive mutations? Well, this is where the power of duplex sequencing lies. In the next step, you also form a consensus sequence between the two single-strand consensus sequences. So the duplex consensus sequences are formed, and then only if a mutation was present in both strands of your DNA molecule, it is called a true mutation. And by using this approach, very low mutation frequencies can be analyzed. The error rate of this method is lower than 10 to the -7. In the originally published method for the consensus formation, they were relying on first mapping the reads to a reference sequence, and only after this step the consensus sequences were formed. But when working with very low DNA input amounts, you want to recover as many sequencing information as possible from your sequencing run. And by doing this initial mapping to a reference sequence, you can lose sequencing information at regions which are, for example, hard to sequence and have many sequencing errors, or also when it involves insertions and deletions. And this is why Nick developed a reference-free sequencing analysis or consensus formation pipeline. So next slide, please. Which is the de novo pipeline. And so here you prepare the libraries as described before, but when forming the consensus sequences, you before mapping them to a reference sequence, you first form the consensus sequences and only then map them to the reference sequence. This has the advantage, as mentioned before, that you recover more sequencing reads, because when performing the duplex sequencing, you have to consider that for being able to reliably call variants, you need to have at least 3 initial PCR products for each strand sequenced to get informative information on variants. And this means that for a duplex consensus sequence, you need at least 6 paired end reads that you can get the sequencing information of one initial DNA molecule. And this means, of course, you need a lot of sequencing reads to get info on a lot of DNA, and you want to recover as many sequencing reads as possible. What you also have to consider is that sequencing and PCR errors cannot only happen in your DNA of interest, but also in the barcode sequences themselves. And if you rely on exact matches of those barcode sequences, you would discard a lot of information present in your sequencing run. So this is why Nick also implemented the option of error correction for the random barcode sequences, which allows you to recover more sequencing information with the de novo pipeline. So in the next part of the webinar now, Nick will explain you more on how this de novo pipeline works and also show you how to use it on Galaxy.
Okay, thank you, Barbara. Covered a lot of information on this technique, which is a really powerful one. Which is why I developed de novo, and I'll show you how to use it in Galaxy. All right, so this is an overview of the de novo workflow and the Galaxy tools and how they correspond to the steps that Barbara just talked about. So the core of de novo is 4 tools here. And then there's one additional step where you filter the output. So this workflow takes as its input just regular reads, the raw reads that you get out of the duplex sequencing method. So the fast cues straight from the Illumina machine go in. And then at the end, what you get out is also fast cues, except this time they are duplex consensus sequences, which are, extremely accurate and very reliable. And then you can use those in your variant calling, much like you would standard fast cues full of Illumina reads, except they're much more reliable. So I've included, I'll note there's a bit.ly link down here at the bottom of each of my slides where this links to a tutorial that goes through all the steps I'll talk about here, including also variant calling and a lot more information. So you can copy that down anytime you want. Okay, so let's take you through it. Here's the first tool. It's called Make Families. Basically, it takes your FASTQ files that you got out of your Illumina machine or whatever. It takes each sequence, each read, chops it up into its parts. So here, the first 12 bases are the random tag. And then you got your 5 base, this is an invariant sequence that's used to link the two. And then there's your actual sample sequence at the end here. So this tool will take out the 12 base tag at the start of mate 1 and then also mate 2 in each pair. It'll join those two up and create a 24 base barcode. And this is what it will then use to sort all the reads and group them together into families where they all have the same barcode. Okay, so let's show you how this happens in Galaxy. So here's a Galaxy instance where I've preloaded, here's some raw Illumina FASTQ files. And so then you'll go over to the tool panel here, select the de novo section on most galaxies. De Novo will have its own section here with all of its tools. And you can select Make Families, tell it which of your files are the inputs, the two FASTQ files. And then there's just two parameters, which you'll usually leave as their defaults. A standard duplex sequencing protocol, the random tag is 12 bases, the linker is 5 bases. But if you're doing a variant of the protocol, you can change these. And then we'll just hit execute. Okay, and while that's processing over here, let's go and look at the next step. Okay, so here's the correct barcodes tool. This is technically optional, but I recommend it as it can really help increase your final yield. So it does this by correcting errors in the barcodes. So the way it does that is basically by aligning all the barcodes to themselves and matching up similar barcodes that way. And so it can find barcodes that differ by up to 3 substitutions using this method. So as a little demo, a little visual here, here's a toy example where there's let's look at this family where there's yellow and blue tags. Over here, there's 2 yellows here, and then a slightly shaded yellow, which I'm indicating that there's an error in that barcode. Right now, the tool doesn't know that these are actually similar barcodes that just differ by an error. But if you're able to correct that error, then over here you have enough reads to call a consensus sequence. So instead of all of these reads being wasted, you're now able to use these and get that information, which is pretty cool. So, all right, let's show you how you do that in Galaxy. You select correct barcodes over here. The input is just the output of the last tool, which, by the way, all of the outputs are simple text files, and their formats are described in the text below each tool. So there's a couple parameters here. The important one I'll talk about is the maximum number of differences. So here, the default, it's set at 3, which is sort of the maximum strength correction. This is how many substitutions will allow between barcodes. You can set it between, 0 and 3. 0 wouldn't do anything, but the only trade-off here, 3 will get you as many corrections as possible. The only trade-off is that it's possible to erroneously join up barcodes which are not actually from the same family. They just happen to be close, but at least in our simulations, that is very rare. It only occurs 0.3% of the time. So I usually leave this at 3, and then you get the maximum amount of error correction. So I'll make a quick note here that if you'd like, you can hit yes to send usage data back to us, which can help with development of the tool. It does not include any sensitive information at all. You can read, it talks about it all here, but if you'd like to help us out, you can do that. The default is no. Okay, so then you hit execute. And while that's working, let's talk about the next step, which is multiple sequence alignment. So, we've grouped all our families together. Now we want to call a consensus sequence for each of them, but first we need to align them. So let's go back to Galaxy, select Align Families. And there's not many parameters here. K-align2 is a good aligner. It's the default and it's fast. And let's hit execute. Okay. Now we get to consensus calling. And this is sort of kind of the heart of everything in one sense. You're going to call your single strand consensus sequences and then pair those up and call your duplex consensus sequences. So let's step into that a little bit. And so here's an example, a toy family aligned together with some differences highlighted. And the first thing that de novo will do is it'll look at the quality information that you got from the FASTQ file. And it'll look at the Phred score of each base. And if the Phred score is below a threshold, we'll just ignore that base in the consensus calling. So this is the main place where de novo deals with quality information of the input. So here in this example, we've masked out a couple of bases here. And then let's talk about how the consensus calling works a little bit. So here's the final output, and let's look at, over here, this C came from this column where we have 4 C's and a T. Now, as Barbara mentioned, we like to see our consensus base in at least 70% of the reads. So this is a 4 out of 5, that's 80%. So this looks good. We're pretty confident this is a C. Now over here, there's 3 T's and 2 A's originally, which, if you're using a lower threshold like 50%, you might call that, T's in the majority here. But these 2 T's are low quality, and so we're just going to be ignoring that. And now we're left with 1 T, 2 A's out of 5 bases, 5 reads. So there's no, nothing's above the 70% here. So we're just going to say, we can't call that. So we use N over here. Similar situation over here. 2 A's, 2 C's, a G. No clear winner. So now we have a single strand consensus sequence. And next step is to bring in the other strand, the consensus sequence we called from another family. We align them up and look at whether they agree with each other. So here I've highlighted a place where we called a G in this strand, but a C in this one. So, maybe early in PCR, maybe the first round of PCR, there was an error. One strand got a G, one got a C. So we've identified that. We don't know which is correct. We only have 2 instances here, 2 votes. But the important thing is we're not going to mistakenly, if we only saw one sequence, we'd think, oh, this is a G, and trust it. But instead, we know that, there's something going on here, so we're not going to call it as either. And in the double strand consensus sequence, we use an IUPAC ambiguity code to say it's G or C, so it's an S. And then similar over here, N, A, we're not sure. We just say it's N. So now we have our double strand consensus sequences. And let's show you how that works in Galaxy. Let's make consensus reads. Take the input of the last tool. Here, is our threshold. We want to see 3 reads in a single strand family before if we're going to call anything. Our threshold, if you want to be less conservative, you can use the default 50, but best practice is 70%. So that's saying we want to see our consensus base in 70% of the reads. And then down here, we'll tell it to make FASTQ. So the output will be regular FASTQ files. The only note is that there's currently no good way to assign Phred scores to these consensus bases. So to make a valid FASTQ file, we have to just use a dummy score. You can change it here, but if you use a high score like 40, well, first of all, these are duplex bases, so they are pretty accurate. And also this ensures that you don't have bases, just being discarded by downstream tools. Again, you might change this depending on what you're using downstream. And then here is the Phred score threshold that I was talking about earlier. I usually use 25 just to get rid of the real low quality ones. And now there's also an option here to say, give me the single strand consensus sequences too. So this will create two extra files, two FASTQ files, which contain the intermediate single strand consensus sequences, because that can be useful sometimes. Okay, that's it. Let's hit execute. And back to here. So, all right, so this is great. We have our very accurate double strand consensus sequence. It's in a FASTQ file. We can use it just like any other read, but except it's got these weird bases. Some downstream reads, some downstream tools may not like that. Also, you can run into areas where there's a lot of ambiguous bases. Some will not be ambiguous, some will be called. But in those areas, that's a sign that we're not getting a lot of good information. And it's probably best to ignore those areas. So we include another tool called the sequence content trimmer, which basically goes through each of your reads and lops off areas with too many ambiguous bases. So let's show in more detail how that works. Okay, so here's a real read. Basically, the trimmer goes through the read, sliding a window. You can configure all these options, but here it's 10 bases. And if there's too many ambiguous bases in the window, it'll start trimming. Here, our threshold is 20% ambiguous bases in the window. So we got 2 ends out of 10. That's exactly 20%. So we're good here. It'll just keep moving, sliding along. And then it gets to an area like this, where this window clearly has more than 20% ambiguous bases. So at this point, it'll go back to the first one and start chopping right there. And then it'll produce this read at the bottom, where you have some ambiguous bases, but the real bad parts are chopped off. Again, you can configure this if you want to eliminate everything that's not ACGT. You can set that. But over here, I would recommend at least removing areas like this. These figures are pretty good for making sure you just get rid of areas that are just, there's something not great in the consensus column there. All right, so let's look at this in Galaxy. So you select the sequence content trimmer. We're using paired reads. Let's look at the double strand consensus sequences. And here the default is to say, here, these are the bases that we want to filter out. The default is to say get rid of all the ends. But let's say I just want to get rid of anything that's ambiguous. So go down here, invert the meaning of that field. And now these are the bases we want to keep. So let's say ACGT, and anything else we want to filter out. Here is that threshold where this will mean now 20%. So we'll start trimming at windows with 20% of these bases. Size of the window is 10. And down here, we can set an option where, you know, if you run into a situation where there's a, you know, bad, you know, it's the reads just start getting bad really early on, you know, you end up with a read that's only like 10 bases or something. It's not very useful for most applications. So here we can say, I only care about reads that are at least 50 bases. And you know, you can change this according to your usage. But I think this is, you know, this is a good example. Okay, so this is all set. Hit execute. And there you go. These are going to be some FASTQ files with our trimmed duplex consensus sequences, extremely reliable reads. You can use them in your, you can go on and do variant calling with them in the tutorial that I linked. You can see how to do that. And you can pretty much use them just like you would regular reads, except, you know, if you're doing variant calling, you can change your thresholds perhaps, because, you know, even if you see a variant in a couple of these reads, of these consensus sequences, you might have something real there, because, you know, these are consensus of several reads on each strand, very reliable reads. So it's a pretty useful method. All right, so that's it for me. Here's de Novo's publications if you need to cite it, and I will hand it back to Barbara.
Yeah, thank you, Nick, for this nice explanation of de Novo. And now in the last part of the webinar, I want to show you an application of this de Novo pipeline in combination with duplex sequencing to detect rare variants in mitochondrial DNA in single oocytes. And if you could go to the next slide. Well, first I want to start with some background on mitochondrial DNA. Well, mitochondria have their own little or short genome, which is a circular genome at the size of 16.3 kilobases in mice. And in comparison to nuclear DNA, mitochondrial DNA, there are around hundreds to thousands of copies of mitochondrial DNA in somatic cells, and even around 200,000 copies of mitochondrial DNA in oocytes. And considering that mitochondrial DNA is maternally inherited, when you want to study germline mutations, oocytes are of a particular interest. Next slide. So mitochondrial DNA can be heteroplasmic, which means that there are more than one copies of, more than one variant of mitochondrial DNA present either within a cell, tissue, or an individual. And those heteroplasmy frequencies can be anywhere between 0 and 100%. But with the majority of the available methods, only variants which have a frequency of at least 1% can be accurately measured on the whole mitochondrial genome level. So because of this, very little is known about what happens at lower frequencies, and especially what happens in the germline. This is why we decided to use duplex sequencing to directly measure de novo mutations in the germlines, where we used oocytes, and also in somatic tissues. And additionally, we analyzed the transmission or inheritance of low frequency heteroplasmies in this work. Next slide. And when working with single oocytes, despite having around 200,000 copies of mitochondrial DNA, you have to consider that they have a very little amount of generally a very little amount of DNA. And in the originally published paper where they were describing duplex sequencing, they required to start with at least 100 nanograms of DNA with for library preparation. But in single oocytes, you have less than 5 picograms of DNA. And so it was very important to recover as much sequencing information as possible from the sequencing results produced from the single oocytes. And this is why de novo was so important for this project, as demonstrated here when using the original reference-based analysis to analyze 300 times 300 base pair reads from a single oocyte. Without trimming of the reads, we could only get 9,605 duplex consensus reads. When trimming the ends of the read, which are usually of lower quality, it could be increased to around 43,000 duplex consensus sequences. But with de novo, without doing any trimming, we could even get 61,079 duplex consensus sequences. And in the table below, it is just highlighted why duplex sequencing outperforms other standard sequencing method in terms of variant detection. When you're interested in low frequency variants, we compared it to oocytes that were amplified by whole genome amplification, followed by TrueSeq PCR-free sequencing. And even when looking at variants at frequencies with a cutoff of frequencies higher than 0.5%, while there were no mutations detected with duplex sequencing, there were 18 false positive mutations with the TrueSeq library preparation. Next slide, please. So in this work, we sequenced 2 independent mouse pedigrees, where we sequenced brain and skeletal muscle as somatic tissues of 36 mice. And those were either mothers at an age of 10 months when the samples were collected, and or pups at an age of around 20 days when the samples were collected. And in addition to those somatic tissues, we also sequenced 92 single oocytes and also 24 pools of oocytes. Next slide. Next slide, please, Nick. Thank you. So now, sorry, the previous one. Could you go back, Nick? Okay, thanks. Here this just shows an overview of the workflow that was used to sequence single oocyte and somatic tissues of the 2 mouse pedigrees. So first, the samples were isolated and oocytes were lysed, or the DNA from the tissues was extracted. We enriched for mitochondrial DNA, and analogous to what I was the beginning, we prepared duplex and sequencing and formed the consensus sequencing sequences using the de novo analysis workflow. And after preparing forming those consensus sequences, we mapped the duplex consensus sequences to a reference sequence and called and analyzed the variants. So next slide. This here just shows the workflow that we used for forming the consensus sequences in Galaxy. And since my background is in wet lab biology, this was really helpful that I did not have to deal with all the single tools and codes, and I could just create a workflow and apply it to the different data sets that I generated. And as Nick also mentioned, you have the option to also get information about the single strand consensus sequences, which I used for some specific applications in my analysis. Next slide. And so as mentioned before, when preparing libraries from single oocytes, the input material is very limited. And after enriching for mitochondrial DNA for oocytes in mothers, the median sequencing depth that we could get when starting with the whole oocytes was only around 350x in the mothers and only around 80x in the pups. Next slide. So one thing that you have to consider when having such low sequencing depths and when you are interested in very rare variants is that if you have overlapping reads, a mutation which is only present in one initial DNA molecule would be counted as 2 mutations. In the majority of other methods, you just discard variants that you find only in one read, but because of the power of duplex sequencing, you can include those variants in your analysis. And so, for example, as shown here, if you do not any further processing, the sequencing depths would be, mutations would be counted by a variant caller. So what we did in this work is, next slide. We clipped the overlaps so that those regions were not counted twice, and that way, for this example, the sequencing depth is only 6x, which represents 6 initial mitochondrial DNA molecules and only 1 mutation. And in comparison, without this clipping, the allele frequency would be 25x, but with the clipping, it would be 16.7x. So, of course, when it's only present in one read, you do not get a reliable frequency, but this is analogous also when it's present in only a few of the reads. Next slide, please. So we use the naive variant caller for calling the variant in this project, which is also available on Galaxy, but there you can also use alternative variant calling tools such as LowFreq, which allows you to specify also more options for variant filtering and so on. Next slide. Yeah, so one major focus of this work was to analyze de novo mutations. Next slide. And with this de novo sequencing, we were able to measure mutation frequencies lower than 5 times 10 to the minus 6 in the PAPs in somatic tissues, as well as in single oocytes and oocyte pools. And we could show that in a span of only 9 months, the frequencies significantly increased in the somatic tissues as well as in the oocytes. Next slide. And so we were also interested in how those mutations were distributed, and for mitochondrial DNA, the D loop is of special interest because this shows a higher number of variants. And if you go to the next slide, please. What we could show in our results is that while in the somatic tissues, there was a significant increase in mutations in the D-loop region, in oocytes, we did not observe a significant increase. So this hints that different mechanisms are involved in the formations of those variants. And since we could directly measure mutations in single oocytes, we were not relying on measuring or on sequencing mother and child pairs, and that way we could get a nice number of mutations that allowed several different kinds of further analysis. Next slide. So one thing that we could also see is that the majority of the mutations were of really low frequency. Most of them were only occurring in one mitochondrial DNA molecule, and also they were mainly at frequencies lower than 1%, which would not be measurable with standard methods. And next slide. So this was for somatic tissues, but also for germline mutations. The majority was at rather low frequencies. There were also some at higher frequencies, which is related to the way how mitochondrial DNA is passed on to the next generation. Next slide, please. Another thing that we could see in our data was that there were mainly transitions accumulating with age, and while in somatic tissues, those were mainly C to D and G to A transitions. In oocytes, the major increase was in A to G and T to C transitions, and so this is also another hint that there are different mechanisms involved. If you go please to the next slide. So this was just an overview on some of the results of this work. And it was recently published in PLOS Biology. So if you're interested in more results, you can read it up there. And finally, I want to thank all the people involved in this project, especially Katerina Markova, in whose lab I performed all this work, and also all the other people involved in this project. So now we are open to questions. So please feel free to ask questions that popped up during this webinar.
Stephanie, you're still muted.
Yep. Yeah. Thank you, Nick and Barbara, for those really great questions. I don't see anything in the chat, but does anybody have any questions that they would like to ask?
Just one, not necessarily question, but clarification. Of course, the example that Nick was using is it's a small set of data, so the analysis runs within the framework of this workshop. But Barbara, you actually analyze much larger data sets, and so you can also you have sort of evidence that this pipeline is robust when you're dealing with very large data sets, right?
Yes. So I used data sets with, most of them were around 8 million paired end reads, but I also applied it to data sets up to around 20 million paired end reads, and it's really robust also for those kinds of data.
Hi, may I ask a question?
Please.
Hey guys. I was wondering, Nick, if in Galaxy, after every step, you would generate metrics that you can plot and use to figure out whether you're doing the steps correctly.
So there's no, there's not a lot of metrics built into the tools. There's some in the sequence content trimmer that it'll print out. I think in the correct barcodes, there's something in the standard error, but I mostly rely on separate tools.
Oh.
Which,
Nick, sorry, you dropped out there. Can you just repeat those last couple of sentences?
Yeah. So actually, there's a lab in Austria, Barbara's old advisor, who they just came up with a whole set of tools which are excellent at getting into the quality of your duplex run, the family sizes, and everything. So they've really done the legwork on that stuff, and it's a wonderful suite. I can post that somewhere, perhaps. Yeah, I'll figure out how to include that. But yeah, that's a good question.
Well, there's a set of tools developed by Irene's lab, so we can perhaps use them.
So my question is, if I see a lot of uncalled positions, basically, how can I tell whether my experiment was wrong in the same place? So that's what I was aiming at.
Yeah. So yeah, it depends on. So there's a lot of. I think there's I'm trying to remember some specific tools, metrics that you get from some of the tools that Irene created. Like the general benchmark I have is, like what I was talking about before, if there's like a lot of ambiguous bases, it's worth investigating. That's what I'll say.
All right,
maybe
great.
one thing, sorry, maybe one thing to what you can check is there's also this check input where you get the basic information on the family sizes of your reads. So because as mentioned, you need at least a family size of 3 per single strand consensus sequence that you form a consensus. And if you do not have enough sequencing reads that for the majority, you would only have, for example, 1 PCR product sequence. This gives you information on this part.
Yeah, that's true. There's a diagnostic tool that I wrote that's included in Galaxy, which you can run before you do all the analysis. But yeah, Irene has really developed the whole suite.
Thank you.
All right, well, if there's no other questions.
I do have another question. I just wanted to give the opportunity to somebody else. This is a question for Barbara, actually. So you can identify very low frequency variants, right? And this is so for if you had paired data, you have a mother and a child, how often do you see very low frequency heteroplasmies in one of them that you cannot, I mean, with regular sequencing, that it is a true positive, but you wouldn't call it as such unless you do duplex sequencing. How often does that happen? So if I have evidence that the allele is present in the mother, in the child, but it's very, very low frequency in the mother, so I don't have the confidence to call it as such with regular sequencing. Would that be, how can duplex sequencing, I don't want to discard that variant because it's very low, right? So why do I need to do additional sequencing to prove that it's real, even though it's very, very low? I don't know if I explain myself.
Yeah, so in my data, I had instances where in the mother, the frequency was lower than 1%, but then I saw it in either single oocytes, sometimes at 5% or whatever, and also in the PAPs at higher frequencies. And so it really depends then on the sequencing depth at what you have sequenced the tissues in the mother. But so one thing that I used here in the analysis is I also included single-strand consensus sequences, because in several cases, it is possible to form a consensus sequence for one of the strands, but not for the other. And so with the duplex consensus sequence, those reads are discarded, but they still already have a higher accuracy compared to standard sequencing. And the sequencing depth for single-strand consensus sequences is usually around 3 to 4 times higher than what you get with duplex consensus sequences. So if the sequencing depth is rather low in the mother, and for example, if I just saw it in 2 molecules in the mother where you cannot really get a reliable frequency of a variant, then I also used the single-strand consensus sequence where it was then maybe present in measured in 8 molecules to get the frequency in the mother. And I don't have a number on how often I observed this in those 2 pedigrees, but it happened. I hope I understood your question correctly.
Yeah, thank you.
Well, I hope that you've enjoyed learning about our de novo method. And I'd like to take a moment and really thank both of our speakers for introducing the method and how to use it. And I hope you've enjoyed our webinar. And our time is up, and I hope that you guys will join us for our next webinar, which will be in January on Oxford nanopore data analysis. So thank you all very much, and enjoy the rest of your day.